I'm going to talk a little bit today about looking at the economy of atoms, and that's a thread that runs through our group, really. So I'm aware that there'll be new people today. So my name is Ger McGlacken, and we work in these areas: catalysis, asymmetric synthesis, CH activation, particularly. We collaborate with Jerry Reen as well on interrupting bacterial communication systems as an alternative to antibiotics. And we work closely with the pharmaceutical industry as well in making some of our chemistry applicable. So I'm going to dive right into pot economy and what it is. Well, it's a very simple concept. It's about using the same vessel to do multiple different reactions and telescoping them through. This can save time, it can save money, it can save washing, and a number of different aspects can be improved if you can do this. It's not trivial though, because whatever residue you have from the first reaction can influence the second one, and especially when you're doing catalysis, because you're dealing with, by definition, usually a 1, 2, 3, 5, 10 more percent catalyst, then whatever you have in there can hinder and can adsorb onto the active sites. And so we were wondering, well, what could we tackle? And aryl-aryl bond, sp2-sp2 formation has been well established at this stage through the Suzuki and Stille reactions, but sp3-sp3 bond formation is still a challenge. And if you don't believe me, maybe you might believe Dave McMillan or Greg Fu, who kind of proposed that this is still an area where we can achieve some strides. And what I'm going to talk about over the next 10 minutes is some really simple chemistry. At least the names will be household names to you in terms of the terminology and the named reactions. But if you scratch below the surfaces, a lot of complexities going on. And I'm going to hint at those at least. And so we started to look at the Wittig reaction, which is obviously a well-known transformation, and reducing down that olefin then in the one pot to carbon-carbon single bond. So that would be formally an sp3-sp3 new bond being formed. And as you can see, this is a well-known and well-established reaction. And I'm going to cut to the chase and just say that it worked really well, surprisingly well. And we try and pride ourselves on doing a substrate scope that is actually applicable in the med-chem space and in industrial circles. Twice during this talk, I'm going to pinpoint some unusual results and give you a little clue about what might be happening in terms of scratching that surface again. But you can see here that the scope is quite large, and you can have quite vulnerable functionality in there, like a nitrile protected silyl protecting groups. But I'm going to focus on this one over here on the left-hand side. So you can see, and I know we have very experienced organic chemists here, of course, but you can see that this surprised us. And maybe you can have a think about why that might surprise you, that this substrate worked particularly, or in particular, this substrate worked for us in our two-step process. And in our paper, we elucidate exactly why that is the case. And it boils down to the fact that we serendipitously poisoned our catalyst in step 1. And because we had it in our same vessel, that poison tempered our catalyst activity in the reduction step. And I can go into that in more detail if people want to chat to me afterwards. And we showcased this one-step Wittig olefination reduction and using a formal synthesis of a natural product. So I'll tell you what's happening here. We're doing a Wittig, then we're reducing the olefin, we're reducing the nitro to an amine, and we're doing a reductive amination, and we're doing a hydro debromination all in the one pot. And catalyzing many of those steps is a very humble, but yet, I suppose, exquisitely complex catalyst that is palladium on carbon. One of the most simplest, at least to write down on paper, catalysts that you will see, one of the most complex in reality. And I see Tom O'Callaghan up there who knows all about the complexities of palladium on carbon when you try and do this in a real system. So this is at least 5 steps all in the one pot with 38% yield, which averages out about over 80% per go. And so that's in terms of pot economy. In terms of atom economy, most people will recognize this guy, Barry Trost, and he developed this concept of taking care of our atoms in every step of a synthetic process. And he termed that atom economy back in the 80s and the 90s. And this is where the SSPC, I suppose, the value added that we found was we collaborated with Lilly to apply for this. It's called an LRAP grant, an internationally competitive grant. And they were interested in looking at reductive amination, again, a well-known process in organic chemistry, and using 1 in every 5 processes to form drugs. And we asked around different pharmaceutical players, what do you use in your reduction step in your reductive amination? And really surprisingly to us, it was these guys. So you've got your superhydride and you've got triacetoxy sodium borohydride. And if you think about atom economy, these are one of the worst examples of atom economy. All you use is this very, very small hydrogen, and then all of these atoms are waste, and they're waste stoichiometric, in stoichiometric waste. And so we felt that hydrogen would be a very useful alternative. We were aware of the problems with hydrogen, but nevertheless, it's a very atom economic reagent. And so again, I won't go through the optimization process in the short time that I have, but we achieved a really nice scope of reductive amination in the reduction amination space, and using really environmentally friendly and benign conditions. So we use 1 atmosphere of hydrogen, palladium PVP. This is the key. So palladium PVP is palladium surrounded by the PVP ligand. And this straddles homogeneous and heterogeneous catalysis, and this is to the core of this work. PVP is used as a food additive. It's very safe. Aqueous ethanol is our solvent, 30 degrees, and we genuinely believe this is the best way to do reductive amination so far. We have this up as a preprint. We haven't published it yet. So again, it works very well. We do the imine in isolation here, but it works well in the reductive amination 2-step process as well. Over 50 examples. Why is it at the interface of homogeneous and heterogeneous catalysis? Well, this catalyst is soluble. That's the first thing. So is it homogeneous? Yes, it is, in that sense. But you also need a surface to undertake hydrogenation or hydrogenolysis. So these are nanoparticles that have a surface but are soluble. Very benign conditions in atom economy, 0.03 mole%. We think it's lower than that, but that's our maximum palladium that's actually used. And you can extrapolate this out significantly. You can do a number of processes in the one pot. It also works to form tertiary amines. I have it on this slide. And you can also use ammonia as well, but as its salt. So ammonium salts, you can use that also. And you can also form tertiary amines as well, which is far beyond the scope of what has been published thus far. We applied it in real case scenarios. So if we look at lapatinib here, for example, we form an analog of lapatinib. This has a number of potentially problematic sites. You can do a hydro-D chlorination here. You have a free NH. You have this quinazoline. And then you have these sulfur, and we all know that sulfur and palladium like to interact. And so we were surprised that this worked so well in a real API type scenario. And using this core apparatus, you can introduce deuterium as well. And this is important for kinetic studies, but also last year we have the first FDA-approved deuterium-containing drug as well. And what's nice about this is it's very difficult to buy a tank of D2. That's going to cost you a lot of money, but you can make D2 in situ. And in our reductive amination process, it can be introduced with really good incorporation. But this is where we think that it might potentially have the most application, so in flow. So the obvious application here would be that we don't have a big tank of hydrogen, so you can produce hydrogen on tap. So we use hydrogen as our carrier gas for our GC, for example. So hydrogen on tap is very, very safe, as long as you produce it as needed. But that's not really the potential advantage here. If you want to do reductive amination in flow, it's possible. You can use one of these cartridges, so it's formerly a heterogeneous catalyst. But the problem is they get clogged, and the secondary problem is that over time your solution will form little rivers to go through your packed bed type system, which then it's not exposed to the catalyst then, and you have significant problems. Whereas if you think of a homogeneous system, you don't have those types of problems. And so again, straddling our homogeneous, heterogeneous aspects, this catalyst performs really, really well. Hydrogen, 1 atmosphere, ethanol to produce our reductive amination product as well. We did a lot of materials chemistry in this project, and we did a lot of computation as well. I'm just going to show you one small part. And this involves using XPS studies, so Justin Holmes, Gillian Collins, and UCC helped us out enormously here. And one of the things we found was after a reaction, we would wipe a lot of the PVP off the palladium. So you can see this from the XPS. So just by reintroducing PVP back into the system, you can recycle that catalyst. So recycle a homogeneous catalyst is not standard. So for the last part of my talk then, I'm going to speak about some of the work that Robin Keogh, who is part of the SSPC, carried out during her PhD. And we're going to look at these heterocyclic systems here and these pendant groups around the core tricyclic structure. So quinoline is a base, but also work with non-nitrogen containing compounds as well. And we thought that we could apply our one-pot approach to add complexity to these basic flat units to try and increase the complexity of the molecules, which is what everybody wants to do. And we use this substrate here, which is a dibromide, and we thought we might be able to do a CH activation direct arylation here, do a HEC, and then reduce down the CC double bond all in one pot. And I'm going to throw out one of these kind of interesting aspects here as well for you to think about. So if you don't have the HEC coupling partner, the cyclization doesn't happen. So if you take out the HEC, the cyclization doesn't happen. Why is that when the HEC is over here and shouldn't really be involved? That's the question. What happens? If you do want to do the cyclization and you want to maintain your bromide, you add in QPhos. Mark Loughton's the world expert maybe on this. You can't answer this question, that's up to others. But this was really interesting. This is really interesting to us. In that what happens here, and when you add in QPhos as a ligand, now you can close this and what's happening. I'm not going to go into that today either, but that's just an aside. But if you want to do the multi-component process, it works really, really well. You use palladium acetate as a simple precursor. We don't use a solvent here per se. We use no phosphine ligand, and we use no inorganic base. So inorganic base is a problem in terms of working up these reactions. It's often used, 1, 2, 3 equivalents. We all know the phosphorus as a ligand has potential problems in that it oxidizes, and then you have to get the p oxide out. But we don't need to do that, and this TBAP is the key to making that process work. And so you can really make very, very complex looking molecules from very simple precursors. So this is simply made from the 4-chloro and the derivatized phenol in one step. And finally then, the last aspect that we're going to look at, again, this was spearheaded by Robin Keogh and two great collaborators here, Amy and Peter. And we looked at some of the products that we were making, and we saw that they were flat, okay? And you might all be aware of that famous, I think it was Taylor paper, Escape from Flatland, turning 2D into 3D. So the Suzuki and Stille reactions have been so popular and so applicable that they've actually determined the shape of the drugs that we now use. And they're too flat. And that means they can't access the three-dimensional space properly. And so one of the things we were thinking about is, could we take our flat molecules and try and introduce a degree of three-dimensionality to that? Because the alternative would be have your three-dimensionality in there from the start and try and do the chemistry, and that's much more difficult. And so we used our humble palladium and carbon again with hydrogen at atmospheric pressure, this time in acetic acid, to, in a late-stage process, introduce a three-dimensionality to these otherwise flat molecules. So here you can harness the power of aryl-aryl bond formation, but then in the late stage introduce the 3D dimensionality. And Robin did a lot more than I have on the slide here, and so did Amy Lowry. But these are just a couple of examples whereby when you form these molecules, these quinoline derivatives, you can very late introduce hydrogen into the system and deuterium into the system to get you these three-dimensional molecules. And there's Robin, who published the middle part that I spoke about just a couple of months ago in Green Chemistry. So that's more or less me finished. I'd like to thank Robin and David Jones, who has worked exclusively on the first part of the project, my current research group, collaborators in materials chemistry, Lily, for supporting us continuously, but especially in the LRAP project. And hopefully in a couple of years' time I'll be coming back to you talking about earth-abundant metals, this time manganese. So we believe manganese sits perfectly in the periodic table to do some of the chemistry that palladium can do, but again it's earth-abundant and non-toxic. And I suppose when I was putting the slides together, I reflected on the SSPC, and you know, it's not too often that if you give an international lecture, you'll get, you know, people from Harvard and Caltech coming up to you asking you, well, you know, tell me a little bit more about the SSPC. How does that work? And I was asked a few times about that over the years. So a couple of years ago, we decided to write a paper on it, and the architecture in the SSPC is such that I think it could be applied more widely even across the world. It's a fantastic, you know, what would you call it, incubation bed to be a part of. And Sarah Hayes did a huge amount of work on this paper. I'd like to thank her specifically. And thank you very much for your attention.